Hi there, this is a short video to explain the process of Western blotting, as this is a main technique used in a study to identify the effect of azelaic acid in inducing apoptosis of nasopharyngeal carcinoma cells as a treatment option. Western blotting is a technique used to identify the presence of certain proteins, specifically when inoculated with certain substrates to identify the effect of expression of certain proteins. Of interest in this article, we are identifying whether the substrate, azelaic acid, will affect the expression of proteins used in the apoptotic pathway, such that if they are increased, we can then potentially deduce that azelaic acid has a therapeutic treatment option in treating nasopharyngeal carcinoma cells. Initially, we have two lines of the nasopharyngeal cancer cells, cis-MPC039 and cis-MPC-BM, which are inoculated with azelaic acid. Following their inoculation, twice they are washed with cold PBS buffer solution. Followed by this, there is the addition of RIPA buffer, which has key ingredients, protease and phosphatase inhibitors, in order to lyse the cells open, or in other words, to break them open and release their contents, such as their DNA, their ribosomes, and many other contents. This is important as the gel electrophoresis step, which is next, enables the DNA and the proteins to be separated by the addition of sodium dodecyl. Following this, once the DNA and proteins are separated, they are transferred onto a PVDF membrane. This is known as the Western transfer. The addition of 5% non-fat milk is added to the PVDF membrane in order to fill up any gaps that are there, such that when we inoculate with the antibodies, this ensures that the antibodies will only bind to the proteins of interest and not to any gaps. This increases the validity and the accuracy of the results. Following that, they are washed with TBST solution to remove any of the milk. This is time for the antibody inoculation, both primary and secondary. Antibodies are used in order to bind to certain proteins of interest. Antibodies are specific to certain proteins, and these are commercially bought. A primary one is used to initially bind to the protein of interest, and a secondary one is used to bind to the primary antibody to ensure its presence. The secondary one is tagged with something to enable the experimenter to see whether the primary one is there. Initially, primary antibodies are added. There is an antibody for certain receptors, in our case, apoptotic proteins, for example, ERK1 and 2, JNK1 and 2, P38, FAS, AKT, cleaved procaspase 3 and 9, and even phosphorylated forms of these proteins, as this informs apoptosis presence. Following this, it is washed with TBST, such that if the primary antibodies did not bind, they would simply wash away. However, if they did bind, a secondary antibody will come in and will actually bind to the initial primary antibodies. The secondary one is tagged fluorescently or in any other sort of way to identify its presence. Following this, it is washed with TBST solution, such that if no antibodies remain, they are washed out. Following this, we can then identify based on the secondary antibody if fluorescence is present, such that it will fluoresce or it will light up based on the fluorescence that was present or the imaging technique that was present on the secondary antibody. It is important to note the primary and secondary antibodies need to come from different animals to ensure that the secondary one will bind to the primary one in an accurate manner to show its presence. Overall, in identifying this, antibodies are extremely useful in identifying the presence of certain proteins. By identifying the proteins, such as the apoptotic ones, we can identify the effect of azelaic acid in expressing these and therefore in inducing apoptosis. Additionally, a control is also used. A control is used in the form of no azelaic acid, such that we can see whether these apoptotic proteins are upregulated depending on the presence of azelaic acid or not. This also informs and enables reliable results to identify whether azelaic acid is really having an influence. Thank you.